So our next talk is going to be by Dr. Partho Sarathy Roy. Dr. Ray is an associate professor in biological sciences at AIIMS Kolkata. He did his PhD in molecular virology from the Indian Institute of Science, and he went on to do a postdoctoral fellow in the Lerner Research Institute in the Cleveland Clinic. And then he became an associate professor at AIIMS Kolkata. His work is mainly focused on RNA binding protein and miRNA mediated regulation of gene expression. And he also works on computational and experimental biology for gene expression networks and drug design. So without further ado, I'd like to welcome Dr. Partho for his talk.
Okay, thank you, Prajwal. And am I visible or audible?
Yes, sir.
Okay, fine. Thank you. So thanks to the IGM teams organizing this All India IGM meet this year. Means I know this is a sort of a abnormal year, and the teams are not being able to meet each other face to face, which is a sad thing. But I think this is a very good effort of coming together and exchanging notes uh, and seeing ways to help each other. So I have been involved with the IGM projects in India from the beginning. Basically, the first year that IGM Kolkata team participated, and since then I have been sort of part of the IGM community, as the IGM organizers like to call it, and I am very happy to see this enthusiastic teams this year which are participating in this All India IGM meet. So in the IGM work, mathematical modeling has come to play an important role. Basically because I think mathematical modeling allows to plan things well and then go ahead with doing some experiments about it. And mathematical modeling is because of the interdisciplinary nature of the teams which are there with people from physics, maths, computer science and engineering. It's also good to do mathematical modeling of this synthetic biology networks. So I will talk of one such work which we did in my lab and which started off as sort of a fun project. You know, like I like to do some fun projects in the lab together with the major projects which go on. And I've seen often the fun projects give rise to the most interesting insights. So I'll talk about one such work where we have actually used mathematical modeling in a way which is somewhat different from the way modeling is actually done. So mathematical modeling is usually done to describe pathways or networks. So they are descriptive modeling. What we have tried to do is sort of a mathematical modeling as an aid to experimentation in a way such that it can actually give rise to biological discovery. So that is something which I'm going to talk about today. And I'll start off by sharing the screen, and let me see if it's so. Is this visible? The screen is visible?
Yes, sir.
Yes, sir.
Okay. So the title means it's the original title of the works, but don't bother about it. So I call it as mathematical modeling for molecular discovery. So basically where we have actually tried to understand a very important network in biology, which involves which is very important in cancer biology, which involves this gene called P53, which is the major tumor suppressor gene in the human genome, in the mammalian genome. And we have used this sort of approaches to see how this network behaves under DNA damage, because DNA damage is one of the major things which can cause cancer. So I'll start off by asking this: what is common among the following things? The World Wide Web, internet, on which we are talking, social media, public transport, scientific collaborations, the human body, genetic interaction. So things from various different types, from science, from society, from social media. And the common thing which connects all of them is that they are all functioning as networks. So because they are composed of nodes, which can be individuals, which can be bus stations, which can be scientific labs, which can be organs of the body, which are connected to each other, okay, by some sort of relationship, which we called as edges in the language of network biology. And networks have been of interest for a long time. And the first very interesting background of studying networks came from this Leonhard Euler's paper on the 7 bridges of Königsberg, which was published in 1736, where he actually tried to look at the city of Königsberg, which was that time in East Prussia, which is now in Russia. It's called Kaliningrad. So there, actually, the city had 4 land masses, which was divided by this river, and there were 7 bridges on this river. And he came up with a problem that whether you can go from through all the land masses, crossing all the bridges once only. Okay, and the simple answer to this problem is no, and he proved it. Leonhard Euler proved it, that it couldn't be done by crossing one bridge once only. And then it was found that this could be solved easily by considering all of the land masses as these points, and then the bridges as this connecting edges. So this is how this entire thing, this geographical mass with the river, bridge, and the land masses was reduced to a network, and this gave rise to the studying of networks as an interesting field of science. So this gave rise to this field called graph theory in mathematics. And then because this was found to represent various things in nature, this became a sort of a very important part of studying networks or things as networks. So that is what we tried to do. And I always was inspired to do this because the way in which we have studied biology, and unfortunately the way in which still most of you are taught biology, is what I call as a one signal, one gene response paradigm. So where we talk in detail about one signal which is coming, and then we talk about one pathway which everyone studies about in detail, and then the expression of a set of genes or one gene in response to that signal. But we know that this is not how things actually happen in nature or in life itself. So at any point of time, any gene in your body or my body is exposed to a multiplicity of signals. You know, like for example, now everybody is sitting in their homes or their rooms, and they are being exposed to the light, to the sound, to my blabbering, which can put you to sleep or might keep you awake. So every gene in your body is being exposed to all these signals, and they're all activating different possible signaling pathways. And the gene, whether it will finally be turned on or off, will depend on the integration of these different signals. Now, this integration will follow laws of mathematical integration, but at much more a complex scale. Okay, so we thought that, and the same thing happens in synthetic biology pathways also. So when you are studying the expression of one, for example, an enzyme, or the activity level of one enzyme in a E. coli or in some other cell, then that enzyme would be regulated by a multiplicity of pathways and a multiplicity of signals. And often synthetic biology projects fail because people just study immediately the upstream enzymes of that pathway, and then when they actually then try to see it, they try to do it in experimentally, they find that something else is getting affected, okay, some other unknown molecule or some other pathway which is affecting the activity or the production of that enzyme. So in synthetic biology, therefore, it is important to study these pathways of synthetic biology of enzymes as networks, and to move away from this one signal, one gene response paradigm which has dominated biology for a long time. And that is what we try to do. And we try to do, taking this very important but very simple sort of genetic regulation process, which is mediated by P53. So P53 is a transcription factor which activates transcription, okay, of genes, and it is exposed and it mediates a cellular response to what we universally call as genotoxic stress. So any type of stress which can cause DNA damage. This can be chemical agents, okay, it can be hypoxia, it can be UV radiation, it can be oxidative stress such as reactive oxygen species, it can be exposure to ionic radiation such as gamma. All of this which cause damage to DNA will cause activation of P53. So P53 will get activated, and what it does it immediately causes cell cycle arrest because it prevents the cell with the damaged DNA from further dividing, and it uses that opportunity to do DNA damage repair. And if it fails to do that, so if the damage is so extensive, it will cause apoptosis of the cell. So the expression of P53 has to be tightly related because it protects the cell from damage. On the other hand, it can also kill the cell by causing apoptosis. So P53 expression is found to, in response to DNA damage, is, and people have studied this, and people have studied using very a very basic network of regulation. So this is shown over here in the left, where when DNA is damaged by UV radiation or gamma radiation, it causes activation of P53 by basically preventing its interaction with another protein called MDM2. MDM2 is a protein degradation enzyme. It's an E3 ubiquitin ligase. It normally causes degradation of P53 and keeps the P53 level low. When DNA damage happens,
Okay, I think Doctor Ray has some internet problem. We'll just get in touch again.
You're not audible,
Hello?
sir. Yeah, we can hear you now.
Is it fine now? Hello? Am I audible now?
Yes, it's better, sir.
Is it okay? Is it okay now?
Yes, sir. You're
Yes.
audible now.
Yes, sir.
I don't know what. I think I got disconnected. Okay. So anyway, so basically, so DNA damage causes activation of P53. P53 in turn causes activation of MDM2 by transcription, and MDM2 then goes and degrades P53 again, and this constitutes a negative feedback loop, which is a very common network motif, as we call it in nature, negative feedback loops. And this results in this sort of pulsatile expression of P53, which you can see in the graph here. So P53 expression goes up, and as MDM2 level goes up, it causes the degradation and therefore the degradation of P53. So P53 is expressed in these pulses in response to either UV or in response to gamma radiation. Now the question is that this is not the only way of regulation of P53. We know that, and work in our lab and other labs have shown that P53 is also regulated at the level post-transcriptionally. That means at the level of translation. Okay, so it is not just protein stabilization, but also translation, which can increase the expression of the P53 protein. And this happens at the level of the P53 mRNA. So P53 mRNA is a target of multiple RNA binding proteins, which you see in all this ellipses over here, and also a number of non-coding RNAs, which can either enhance or inhibit the translation from the P53 mRNA. Okay, and two such molecules on which we have worked in the past is this microRNA called MIR125B, which inhibits the translation of P53 by binding to a site in the 3' untranslated region. Okay, and then there is a protein called HUR, which again also binds.
Sorry to interrupt.
Yeah.
But I think you're not sharing your screen. The slides are not visible.
Is it? I can wait a minute. Oh, I think I got disconnected and then the share screen thing went off. Okay, is it there now?
Yes, sir.
Okay. So basically, now what I was telling is that, so the P53 mRNA translation is regulated by all these molecules which are present over here, which are either proteins or this non-coding RNAs. And two of these proteins and RNAs which actually we know functions is that it's microRNA called MIR125B, which binds to the P53 UTR and inhibits its translation. On the other hand, there is a protein called as HUR, which also binds to the 3' untranslated region and enhances translation. So they work antagonistically to each other. Now, we have shown in the past that these two can actually compete with each other and regulate P53 mRNA translation. So what we therefore did was to try to model this P53 gene expression network using a very simple, nonlinear differential equation. So basically, this is how this networks look like. So here, this is P53, which activates MDM2, and MDM2 inhibits P53. So they are shown by this two arrows, and these are connected by these terms. Okay, so what are these terms? So if we look at the differential equation, we will know what these are. So differential equations describing gene expression are these very simple terms where alpha is a term expressing the rate of synthesis. So this is alpha P53, which represents the rate of synthesis, and beta P53 is a rate of degradation. So any protein, the change over a certain time would be represented by the rate of synthesis and the rate of degradation. Okay, and in between are a regulatory terms where we can represent the effect of any protein on the expression of that particular gene by representing this by this sort of a function, where KMP represents the relationship between these two genes or these two molecules. So KMP, for example, here represents a regulatory constant which represents the effect of MDM2 on P53. Similarly, KPM represents the effect of P53 on MDM2. So we can form these very simple differential equations, and as we can increase the types of regulation on this molecule, we can just keep on increasing these regulatory terms in this differential equation. Okay, so any term anything which will enhance the expression will come as a positive term, and anything which will repress the expression would come as a negative term or as a denominator. So this gives us a very easy tool to mathematically model this type of gene expression networks. So what we do then is to solve these differential equations, okay, numerically, by putting in actual parameters or actual numbers for these different terms, which we can either obtain experimentally or obtain from the literature. And this will give us an expression pattern of that particular gene or genes in response to that particular signal. So that is what we do over here. And here we get an expression pattern of P53, this line which is the computed expression pattern. What we can do now as an experimental lab is also check the actual expression pattern of P53 in response to that signal and see that whether it matches with this computed or modeled pattern also. If it matches that our model is nice, if it matches what is happening in nature. If it doesn't match, it can mean one of two things. Either one is that these parameters are wrong. We have got wrong parameters, therefore it is not matching. And then we can try to change the parameters or refine it and try to see whether it matches. The other more interesting possibility is that there might be unknown mediators in the network, which we are not even aware of, which we haven't included in our model, and therefore we are getting a deviation from the actual experimental data. And then we can now do a further experiments to find that particular molecular mediator. And our work becomes easier now because on the model, we can now guess what sort of a molecule that will be. So we can keep on doing this reiteratively. So what we did over here, so once first we do this modeling, we find that, okay, so this is actually that goes up from 0 to 2 hours, then goes up to 2 hours, and then goes down gradually. So it sort of is with the actual sort of data which we have seen, the computed data, and it also matches with what is known in literature. So we were happy that, but now we molecules, that is HUR and MIR125B, they also regulate p53 expression, because that is something which we have seen experimentally. So, as 25 is a negative regulator, and then
So I think we lost you again.
yeah, I think I had again gone off. Am I back again?
Yes.
Yes, sir, you're back.
So maybe you can try switching off your camera. That might.
Okay. So yeah. So what we now do is we include MIR125B and HUR as regulators over here, and we put in terms for the regulation. We put that in this differential equation. So as you can see over here, we have included the MIR125B as a negative regulator of p53, so it is in the denominator, and we have included HUR as a positive regulator of p53, that's why it is in the numerator, and then we again solve this equation. Okay. And what we now find is that we get a completely different model compared to our experimental data. So we see that P53 now goes up and attains a plateau, and HUR just keeps on increasing exponential, linear. And we see that the HUR expression also doesn't match with our experimental data, because HUR also shows this type of a pulse, which you can see over here. This is the HUR expression pattern. So now, on putting MIR125B and HUR, the experimental data completely differs from the model which is present. So we therefore now try to see that there can be something more. So there can be either a new H, that means a new type of regulation which is present, or a new regulator, that means a new node in this network. So we go ahead. So while we were doing this, another student in the lab independently was looking at MIR125P's function, and he found that MIR125P is actually a negative regulator of HUR itself. So this is the expression pattern of MIR125B. So MIR125B actually, in response to DNA damage, it goes down from 0 to 2 hours, and then it increases like this. And then we found that there was a MIR125B target site in the HUR 3' UTR, and he did a series of experiments. So MIR125 overexpression, you can see HUR level going down. Then we can show that the HUR translation is inhibited when MIR125 is overexpressed. So there's a whole lot of experiments was done, which prove that MIR125B is also a repressor of HUR. So now we put that in the network. So we add a new H to the network, and therefore now we have a MIR125B in this HUR expression differential equation. And now we again do the simulation. Okay, this is just simple doing using MATLAB, and there we now see that okay, the P53 is increasing. It is now attaining a sort of a flat plateau, and HUR was increasing linearly. Now it is also attaining a plateau, but still it is not matching with the experimental data. So there is still something missing. So therefore, so we tried to see now what might be affecting. So there was something which was causing HUR to basically go down, which we see in the experimental data, and we thought that we wanted to check whether HUR was getting degraded. So we did an experiment to see the stability of HUR, and we find that HUR is a super stable protein. So this is using cyclohexamide, which stops protein synthesis. So here then if we look at the protein level at different time, we will see the rate of degradation only. And we find that HUR doesn't degrade at all over a 12-hour period. However, if we give UV, the DNA damaging agent, it degrades nearly completely during the 12-hour period. So something UV was doing which was causing degradation of HUR. And then we tried to see that whether this degradation was mediated by the proteasome. So we used a proteasome inhibitor, and we now see that the HUR is not getting degraded. And then we also did experiments. We showed that HUR was actually getting ubiquitinated during this time, and this was actually leading to its degradation. So we now knew that there was something which was causing the degradation of HUR, possibly during this period. So now we include a hypothetical factor X, which we don't even know about in the model, which we take as a HUR degradation factor, and then we put in the rates because we can measure the rate of degradation, etc. And then we solve this differential equation. So now we have this factor X as in the HUR term as a negative regulator of HUR. And Eureka, what we find now that the modeling pattern, the simulation result now nearly completely matches with our experimental pattern, with the experimental data. You can now see here we see an increase and decrease in the case of the model output, and which matches with that of P53. And again, here we see that it matches with that of HUR. So now, therefore, we have predicted that there is a degradation of HUR which regulates the level of HUR and P53, and it is mediated by factor X. Now, most models will end over here, but we as an experimental lab, we really wanted to see that whether we can find X. Okay, if it is predicted, whether we can find it. So what we did, we took cells, these are breast carcinoma cells, we treated it with UV, with cyclohexamide to stop the synthesis, and Mg132, because if there is something which is degrading HUR, it will bind to HUR, but if HUR degrades, we will not be able to find it. So we stop the degradation of HUR, and then we use immunoprecipitation by using an anti-HUR antibody to pull down HUR and any protein which is associated with HUR. And then we do mass spectrometry to identify these proteins. And what we find is very interesting. We just find 3 proteins in this mass spec result. One is HUR itself, which is expected. And among the other 2 proteins, one is an E3 protein ligase called TRIM21. And E3 protein ligases are proteins which are responsible for degradation. Now, no one knew that TRIM21 interact with the HUR or causes degradation of HUR or was a part of the DNA damage response pathway. People have been studying the DNA damage response pathway for the last 30 years, but still people didn't know that it was part of the DNA damage response pathway. So TRIM21 is an E3 protein ubiquitin ligase involved in regulating inflammatory responses and carcinogenesis. So it is basically one of the E3 ubiquitin ligase which is required to bring down the inflammatory response. So it degrades this protein called DDX41, which is activated by double-stranded DNA, which is present in DNA viruses. So it degrades DDX4 and therefore brings down the inflammatory response. But no role of it in the DNA damage response was known. So we now could do a series of experiments. So we pulled down HUR using HUR antibody, and we could pull down TRIM21. We could do vice versa. We could pull down TRIM21 and we could pull down HUR. We could show that this association between TRIM21 and HUR was enhanced by UV. So here this is in presence of UV. So this increases the pull down. You can also show that this association of HUR and TRIM21 increases in a time-dependent manner in response to UV in the same way that we see the pulse happening. That means as HUR level goes down. We could also show by using siRNA against TRIM21 that if we degrade TRIM21, even under UV condition, HUR does not get ubiquitinated. Although in terms of control siRNA, HUR gets ubiquitinated. So this is the ubiquitination gel. And we can also show that when we use this UV and cyclohexamide, we can show that there is the degradation of HUR by TRIM21. So now the question was that we have seen that our experimental data replicates the model. Now the question is that, and that is the challenge, whether now we can manipulate the experiment and recreate the intermediates of the model. Okay, so basically that is what we have done over here. This is sort of a busy slide, but it's easy to sort of follow if you just follow me. So this is, we have transfected cells with a control oligo. Okay, and here you will see this pulse of HUR and the pulse of P53. Okay, in presence of in response to UV. Then we have used an antagomer to 125B. This is the inhibitor of the microRNA. And here now you can see that the P53 goes up and attains a plateau, and so does HUR. So that is what you will see, that the antagomere, which is the green line, it causes both of them to go and attain a plateau, which is what we have seen in one of our modeling steps. We use TRIM21 siRNA. Again, we see that TRIM21 siRNA causes P53, the HUR to go and attain a plateau, because it causes degradations. If you now inhibit TRIM21, this will attain a plateau, and P53 goes higher. Okay, and then when we use both antagomere 125B and TRIM21 siRNA, then we see that both that HUR increases linearly, which we had seen in our initial model, which had neither mid 125B nor TRIM21, and then P53 also goes up and attains a high, nearly a linear. So basically, every step in our model we can go back and replicate by now. Basically, like inhibiting or manipulating each of the steps of this network now, which we have found. So basically, we have also now seen that whether this can actually affect the function of the cells, whether it actually affects the phenotype, because this is just the molecular level what we have seen, and that is something which we have seen that yes, if we use TRIM21 antagonist 125B and TRIM21 siRNA, remember this will cause P53 to keep on increasing, and P53 keeps on increasing, it will cause apoptosis of cells. So here, therefore, we see that there is a decrease in cell viability. Okay, when we inhibit both mid 125 and TRIM21, and there is, and this is seen in the case of an aggressive breast cancer cell line MDMB231, where this decrease, where actually P53 is absent. So P53 is inactive in MDMB231, and there we do not see any difference over here, which means that whatever we are seeing over here is actually being mediated by P53. When P53 is absent, there is no difference between the control and the treatments of antagomere and TRIM21. So basically, this tells us that starting with this very simple network, which was the only thing which was known, P53 MDM2 regulated by DNA damage, we can keep on expanding the network, enriching the network by putting in new mediators and new edges, okay, new relationships, new interactions, and finally we have come up with this much more complex and much more enriched network, which represents the reality much more. And there we now see there is a negative feedback loop between HUR, between P53 and MDM2, which was known. Now there are 4 different feed-forward loops which is present now, which can also regulate P53, and thereby can regulate P53 levels. And already we now know that there are even further, this is still not the complete network. With this, we can keep on building, because for example, we now already know that HUR can negatively regulate mid 125B by causing its export from the cell. Or recently, we have just published a paper last week where we can show HUR can positively regulate TRIM21 by enhancing its translation. So basically, this is an important thing, and is also important, very, very important clinically, because now we have gone back and seen that till now no one knew that TRIM21 was actually involved in regulating P53, but now we are seeing in many cancers where P53 level is actually low, but that is not because of a mutation in P53. There is no mutation in P53, but we see that there is a mutation in TRIM21 which has caused its upregulation. So therefore, many cases in chemotherapy, when a particular gene or a molecule is targeted, often the chemotherapy doesn't work because possibly the problem is with some other regulator which is upstream of that gene, which we are not even aware of. So this also helps us clinically to develop new therapeutics and a better understanding of the networks which I'll end by thanking the team. This type of interdisciplinary work is only possible if I have a team of very involved and enthusiastic individual. That is what we had. So this work was actually initiated by Vibudha, who was a SMS here in AIIM Kolkata, who was just interested in mathematical modeling, and then he wanted to do some experiments. So he used to hang around in my lab. So he started this as his MS project. So he was a chemistry major, but he did a project in my lab in biological sciences. And then with him worked my friend, who was a who is basically a biochemistry, like a major MSc. And then it was also, the work was done by Krishan, who was a PhD student in mathematics. And then now the work was carried on by Sukhen, who is a computer engineering graduate, who is now doing his PhD in my lab. And Abhishek Gua, who was a zoology master student who just finished his PhD in my lab. And we had as a very close collaborator and consultant Anandamohan Ghosh, who is a professor of physics. So this project had contribution from physics, chemistry, mathematics, and biological sciences, all the streams at AIISR Kolkata, which came together to do this type of work. And also the funding was provided by Wellcome Trust GBT India Alliance, allows us to do this type of very interesting, like new type of work, which we usually don't do in our labs. So I'll end here, and I'll be happy to take any questions or comments that you might have. Thank you.
Thank you, sir. Thank you for your talk. If anyone has any questions, please feel free to go ahead and put it on the chat, or speak out and ask a question. Okay, looks like no one's asking a question. All right, let's join me in thanking Dr. Partho for his wonderful talk. Thank you, sir. Thank you for taking your time and help giving all of us a bit of insight into your work. Thank you.
Thank you.